Hello and welcome. My name is Waqar Hanif. Today I'll be giving a presentation on differential production of abnormal isoforms in brain cells contributes to neuronal death in Parkinson disease, gender-specific differences. So this is a particular study that I've conducted on Parkinson disease, which is common neurodegenerative disorder. It is characterized by the loss of nerve cells, and if the nerve cells are lost, it can lead to impaired brain function by the loss of dopamine production. PD has a significant global impact because it can cause lots of deaths and disabilities. PD affects more men than it affects women because men are more susceptible to chemicals and other material, which leads to multiple folds of risk factor when it comes to PD. Neural death and axonal injury are common features of multiple neurodegenerative diseases, including PD, and apoptosis plays a very crucial role in development of the PD and prevalence of the PD because if there are early apoptosis of neuronal cells, it can lead to neurodegeneration of the brain and impairment of the brain basically. So the idea behind this study was to understand the apoptotic dysregulation in PD, which can help us to identify therapeutic biomarkers that can be targeted using multiple target therapies to prevent the early neuron death. Because if there is early apoptosis, it can cause early neuronal death, and this is a key factor behind this research that I wanted to study how and what sort of genes can cause early apoptosis by their dysregulation. To study this, this is the overview of the study. To study Parkinson disease with that perspective, as I shared earlier, the study was conducted to predict differentially expressed genes and differentially produced genes with the potential to cause early apoptosis in men with PD as compared to women, because in men in PD is more aggressive, and if it is more aggressive, that it means there is a higher rate of apoptosis occurring within the men who have PD. So that is why we should be looking for those genes in men with PD that are causing early apoptosis. I used both RNA sequencing and structural biology analysis techniques in this study to elucidate the genes and their interacting partner that can cause early apoptosis in the neural cell and eventually lead to the death of the cells. So when it comes to methodology, I applied a typical RNA sequencing pipeline, which includes taking the raw RNA sequencing data, which I took from NCBI Geo, and the data was collected from human brain tissue of 18 diseased individuals and healthy control who did not have any neurodegenerative disease. The data was pre-processed, of course, because the RNA sequencing data is usually does not have good quality. So that is why I removed artifacts, primers, PCR primers. I removed adapters and low quality reads before mapping the reads to a reference genome GRCH38 of Homo sapiens using HiSat2, which is a splice aware aligner, very efficient and very fast. And then using the mapped reads, I quantified the reads using string tie, which is a abundance estimation tool, and using after string tie quantification, I used the quantification count table in Balgon, which allows us to do differential gene expression analysis, which was a goal of this study, to identify genes that are differentially expressed between men and women. And then the package was implemented in R, and the thresholds that were used was greater than log FC1 and less than -1, and the p-value was 0.05. So if the gene has p-value less than 0.05 p-value, then it means that the gene is statistically significant, and if the same gene has a value log FC value greater than 1 or less than -1, then it means that it is also biologically significant. So the flow chart is given over here. That means that this is the flow that was conducted. This is the flow of the analysis that was conducted in this research specifically. So then, after identifying the differentially expressed genes, I went on to shortlist those genes that may have a role in apoptosis genes, and the genes were identified through literature, and those genes were specifically shortlisted from the entire dysregulated gene sets, and those genes were specifically analyzed using Isoforms, which analyzer tool in our language. This tool allows us to identify isoforms or alternate splicing in the gene, and these genes were then shortlisted, and those genes were shortlisted that have multiple or more than one isoforms being produced by the same gene. And those genes were then analyzed through functional analysis using gene codis 4, which allows us to do KEGG path analysis and gene ontology analysis. So I specifically chose Go molecule function because I wanted to understand the function of those apoptotic genes. Then then the genes that had potential to cause early apoptosis and direct relation in the apoptosis and are producing multiple significant isoforms, those genes were shortlisted. One of them was TNFSF10, and because it provided the best significant finding and had a direct role in apoptosis, this gene was shortlisted, and the 3D structure prediction was conducted for this particular gene for both wild type, its original sequence, and the isoform that it was producing, that was differentially expressed in men and women. So the 3D structure prediction was done for these both type of proteins. And then to understand if the original wild type protein, which is also of course upregulated, and if the isoform which is being produced by the original gene, do they have apoptosis signaling chances or not? Can they induce apoptosis signaling or not by binding to their original partner, which is death receptor 4? So I did molecular docking using HDoc tool to identify their interacting partners. And then when it comes to results, I identified multiple genes that were dysregulated, namely identified downregulated genes that 332 and upregulated genes were 121. And out of these genes, we identified multiple apoptosis genes, which I'll share later on. So when it comes to when it comes to specifically this volcano plot, on the right hand side the upregulated genes are given in red, and on the left hand side the downregulated genes are given in red color as well. So when it comes to apoptosis genes, these and their isoforms, shortlisted those genes that that had the potential to cause apoptosis were shortlisted, and their isoforms were also calculated using isoform switch analyzer. There were only 5 genes in total that have apoptosis related function and have isoforms in PDB. So in PD, and the upregulated genes were only one, which is TNFSF10. So that is what our goal was, that if a certain gene can cause apoptosis and it is also upregulated, it should be further studied. So TNFSF10 and CASP7 were the only 2 genes that had a direct role in apoptosis, but CASP7 was downregulated. So I chose TNFSF10 because for further analysis, because it has a direct role in apoptosis and it was also upregulated in men as compared to PD. And we also identified a certain transcript, isoform transcript, that was being produced by this gene, which was highly upregulated in men as compared to women. So that should be explored further. So when it comes to functional analysis, molecular function analysis of TNFSF10 revealed that it has functionality in trail binding, tumor necrosis factor binding, zinc binding, etc. And this has the potential to alternate splicing of this particular gene has the potential to cause early apoptosis and may lead to PD. So apoptosis signaling assessment using molecular docking revealed that there are significant interactions between TNF-SF10 isoform and compared to the wild, and a wild protein and isoform protein. So over here what we did was we produced the isoform protein structure and we produced the original protein structure, we superimposed them, and we can see there is a clear difference because the isoform protein was lesser in size, in residue size, as compared to the original wild type protein. And after docking we saw that the wild protein had a -3. The more the value is in negative, the better the value, the good binding. And we saw that the isoform had less value, docking score value, as compared to the wild. So that means that after being muted, after producing a new isoform, TNFSF10 loses some of its functionality because we identified more significant interactions between the original wild type protein and DR4 as compared to the isoform DR4. So when it comes to discussion, apoptosis programmed cell death is a key feature of PD that can affect more males. Splicing dysregulation and isoform variants may contribute to neurodegeneration. And this study focused on TNSF10 gene isoform because it is more prevalent in male and specific PD, as we saw in the results. And specifically molecular docking revealed that there is strong binding between the original wild type protein and the DR4, and that can cause early apoptosis because there is significant binding. And since the gene is also highly upregulated, which we identified, and then when it comes to the isoform that was produced by the same gene, it leads to lesser stronger binding to the DR4. That means it can lead to loss of neuroprotection, because it has to bind to DR4 to properly perform its function. Should not be higher, should not be lesser. So if it is higher, then it can cause early apoptosis. If it is lesser, it will not cause early apoptosis, but it will lead to neuroprotective loss of neuroprotective activity. The study proposes these isoforms along with other dysregulated genes as therapeutic biomarkers, which should be targeted to and that may eventually solve the issue of aggressive PD in male. The study identifies TNF as an alternate splicing gene that is upregulated in PD. The parent isoform hindered the normal apoptotic response in neuron cells. Molecular docking reveals that increased apoptosis and cell cycle arrest mechanism between TNF10 and DR4. On the other hand, if there is weak binding, it can lead to loss of neuroprotective events. This disruption of apoptotic mechanisms in the nervous system may be linked to Parkinson's disease in men, and inhibition of these genes can lead to prevention of early apoptosis in neurons and can help in better management of the PD disease. I hope this makes sense. If there are any questions, please feel free to discuss. I'll be happy to answer them. And I acknowledge the organizers of this event. I also acknowledge Science School at NUST and BioCode for providing the resources. Thank you so much.